Notice something right now, not something outside of you, something happening inside you at this exact moment while you are sitting here watching this. Your body is running a defense operation you have never been told about. No symptom, no signal, no warning light on the dashboard, just a quiet, relentless process working in the background of your biology every single day. And here's the part that changes everything. How well that process works is not random. It is not purely genetic. It is not outside your control. It is being shaped right now by what you ate this morning, by what you had for dinner last night, by patterns so ordinary, so unremarkable that most people never connect them to what is happening at a cellular level inside their own bodies. What I am going to show you in this video is not a wellness trend. It is not a list of superfoods to buy at a specialty store. It is a framework backed by decades of peer reviewed research that explains exactly how specific foods activate three biological defense systems you already have built inside you. Systems that, when properly supported, give your body a dramatically better chance of identifying, containing, and neutralizing cellular threats before they ever become something serious. By the end of this video, you will understand something that most people never learn from their doctors. Not because doctors are hiding it, but because a 15-minute appointment is not enough time to explain it. Stay with me. This is the kind of information that genuinely changes how you think about what is on your plate. My name is Dr. Megan Foster. I have been in surgical practice for 24 years. In that time, I have operated on patients at every stage of illness. I have had conversations that no physician ever wants to have. And I have watched again and again how the biological environment a person builds inside their body over years quietly determines which direction those conversations go. That experience is why I care so much about what I'm about to share with you. Now, let me start with the belief I want to challenge directly. Most people believe that cancer is either something you inherit or something that just happens to you, like lightning, unpredictable, unavoidable. You either get unlucky or you do not. That belief is incomplete. And that incompleteness costs people something real. Yes, genetics matter. Some gene variants raise your risk. That is true, and it is worth knowing. But here's what the genetics conversation almost always leaves out. Carrying a gene associated with cancer risk is not the same as developing cancer. The gene is a loaded possibility. What actually determines whether that possibility becomes reality is the biological environment surrounding it. Genes load the gun. Environment decides whether it fires. And the internal environment inside your body, shaped by what you eat, how inflamed your tissues are, how active your immune system is something you have genuine influence over. More influence than most people ever realize. This is where those three defense systems come in. And I want you to understand them clearly before we talk about any specific food, because this framework is what makes the rest of the video genuinely useful. The first system is immune surveillance. Right now, your immune system is scanning every cell in your body, looking for cells that are behaving abnormally. When it is working well, it identifies those aberrant cells early and destroys them quietly, efficiently, before they can multiply. When it is weakened by poor nutrition, poor sleep, or chronic stress, those same cells can slip through undetected. Not because the threat changed, because the security system got compromised. The second system is inflammation regulation. A short burst of inflammation is healthy. It is how you heal a wound or fight an infection. But chronic low-grade inflammation that never fully shuts off is a different thing entirely. It creates a biological environment where abnormal cells are more likely to survive, where cellular repair is slower, and where the chemical signals that should be triggering cell death start to fade. Chronic inflammation is like a slow fire burning in your tissue. You cannot feel it, but it is doing damage. But it is. The third system is angiogenesis control. Here is something most people do not know. A cluster of abnormal cells, even a potentially dangerous one, cannot grow beyond a tiny size without first building its own blood supply. It needs new blood vessels to feed itself, to get oxygen and nutrients to survive. Your body has a natural regulatory system that controls the formation of those new vessels. When that system is functioning well, even if abnormal cells form, they cannot build the infrastructure they need to grow. They stay microscopic, contained, harmless. Three systems: immune surveillance, inflammation regulation, angiogenesis control. Every food we are going to discuss today influences at least one of those systems. The most powerful influences all three at once. Let me give you one mental model before we start the list because it will make everything stick. Imagine your body is a city, a complex, around the clock city that never sleeps. Abnormal cells are unauthorized construction projects that appear in different parts of the city. Small at first, barely noticeable. Your immune system is the security force. Constant patrols, fast response. When security is well funded and sharp, unauthorized projects get shut down quickly. Chronic inflammation is constant background chaos across the city. Noise, fires, disruption. Security gets stretched thin, some things slip through. And angiogenesis, the blood vessel formation process, is the city's utility grid: water, power, resources. An unauthorized development cannot become permanent without getting connected to that grid. Control the grid, and even projects that start up can never become anything significant. Food is how you fund security. Food is how you calm the chaos. Food is how you control access to the grid. That is the framework. Keep it in mind as we go through each item. Now, before we begin, I want to set accurate expectations. I am not going to tell you that eating these foods will prevent cancer. No honest physician would make that claim. What the research clearly and consistently shows is that dietary patterns built around these specific compounds correlate strongly with lower cancer incidence, better immune function, reduced inflammatory burden, and stronger angiogenic regulation in large, long-term population studies. Studies from institutions like Harvard, studies following tens of thousands of participants over decades. This is not fringe science. It is published, replicated, and increasingly mainstream in oncology research. What you are about to hear is not the loudest thing on the internet. It is one of the quietest and most important. Let us begin. Number 6 is berries. I know, you have heard that berries are healthy your entire life. You are probably already skipping ahead in your mind. Do not. Because what most people think they know about berries is not actually why berries matter. The common dismissal sounds like this: too much sugar, too trendy, not enough impact to bother being intentional about. That dismissal is based on a misunderstanding of what is actually inside these fruits. The story is not the sugar. The story is the anthocyanins. These are the pigment molecules responsible for the deep blue, red, and purple colors in berries. And they are not just pretty. They are biologically active in a way that directly supports the first of our three defense systems. Here's the simple version of what they do. Every day, the normal chemical activity inside your cells produces unstable molecules called free radicals. These molecules are like sparks. Left unchecked, they damage the cellular machinery responsible for accurately copying your DNA when cells divide. Errors in DNA copying are one of the earliest steps toward abnormal cell development. Anthocyanins are remarkably effective at neutralizing those sparks before they reach the machinery. A study published in the Journal of Nutrition involving human participants over multiple weeks found that consistent anthocyanin intake produced measurable reductions in DNA oxidation markers in the blood. Not theoretical reductions, measured ones. The biological effect was real, quantifiable, and appeared within weeks of consistent daily intake. The dose is 1 cup per day. Fresh or frozen, it does not matter. Berries are often flash frozen at peak ripeness, which preserves the compound concentration well. blueberries, blackberries, raspberries, dark cherries. As a general rule, darker color means higher anthocyanin density, the mistake that cancels most of the benefit, berry juice. When you juice berries, you lose the fiber that slows digestion and modulates compound absorption. You concentrate the sugar without the structural context that makes whole fruit metabolically different. And you eliminate much of the anthocyanin content that binds to fiber in the first place. The whole berry is the point. 1 cup daily, that is number 6. And number 5 is going to add something that berries cannot do on their own, a mechanism that goes deeper. Number 5 is pomegranate. The misconception here is that pomegranate is a trend, a fruit that had its moment a few years ago on health blogs and has since faded into background noise. The biology does not care about trends. Pomegranate contains a group of compounds called L-gitannins. When you eat them, the bacteria in your gut break them down into active molecules, one of which is called urolithin A. And urolithin A has been studied for its effects on a process called apoptosis. Apoptosis is programmed cell death. It sounds alarming, but it is one of your most important protective mechanisms. Cells that have accumulated too much damage, cells whose DNA has been copied incorrectly too many times, are biologically supposed to self destruct. They have a built in shutdown code. When that code works, potential threats are eliminated quietly, automatically, before they can become anything serious. In early cancer development, one of the consistent patterns researchers observe is that this shutdown code gets switched off. Damaged cells that should be dying keep dividing instead. They lose the ability, or the instruction, to remove themselves. The compounds derived from pomegranate appear to help reactivate those signals. A clinical study published in Clinical Cancer Research followed men with rising PSA levels, a marker of prostate cancer activity, and found that daily pomegranate juice consumption was associated with a significantly slower rate of PSA doubling time compared to controls. The average doubling time in the pomegranate group was 54 months compared to 15 months in historical controls. Those are not trivial numbers. That is a meaningful biological effect. Whole seeds are preferable to juice because the seed structure itself contains additional active compounds, and the fiber slows absorption in a beneficial way. A quarter cup of seeds a few times per week is a reasonable practical target. If you use juice, it must be pure, unsweetened pomegranate juice. The sweetened pomegranate beverages sold in most grocery stores are essentially colored sugar water. Read the label carefully. That is number 5. We have now addressed DNA protection and cellular cleanup. Number 4 is going to surprise almost everyone. It is purple potatoes. I can hear the skepticism. Potatoes have been nutritionally maligned for so long that the word itself triggers a kind of defensive reflex. And for processed white potato products, some of that wariness is warranted. But here is what that blanket dismissal misses. The color of a plant food is almost always a signal of specific bioactive compounds. Purple potatoes contain a high concentration of anthocyanins, the same family we discussed with berries, but in a form and context that has been studied specifically for its effect on the third of our defense systems, angiogenesis control. A study out of Penn State found that men who ate purple potatoes daily for 4 weeks showed significantly reduced levels of interleukin 6, a key inflammatory marker, and measurable reductions in markers related to blood vessel regulation compared to a control group eating white potatoes. The mechanism that researchers focused on was the anthocyanin interference with certain angiogenic signaling pathways. In plain terms, these compounds appear to help your body regulate the process by which new blood vessels form, including the ones that abnormal cell clusters depend on to grow. Go back to the city model. Purple potatoes are working on the utility grid. Preparation matters enormously here. Baked or steamed preserves the anthocyanin content. Deep frying at high temperatures destroys a significant portion of it. A baked purple potato with olive oil and herbs is genuinely one of the most underrated additions to a cancer protective dietary pattern. Swap it in once or twice a week wherever you would otherwise use a white potato. Now we cross into a different tier. The next two foods do not just support existing defense systems, they influence how your cells read and execute their own instructions. We are talking about gene level activity, and that is where the biology gets particularly powerful. Number three is turmeric, specifically the active compound inside it called curcumin. The dismissal is predictable. It is just a cooking spice, a flavoring. How much biological effect can a seasoning actually have? Here's the answer. Curcumin has been the subject of more than 3,000 peer reviewed studies. It is among the most researched natural compounds in all of biomedical science. And the effect that appears consistently across that enormous body of research centers on a cellular signaling pathway called NFKB. NFKB is essentially an on off switch for inflammation. When it gets activated, your cells start producing inflammatory molecules. A short activation is healthy and necessary, but in many people, due to chronic stress, processed food consumption, poor sleep, and excess sugar, NFKB gets stuck in the on position. The switch never fully turns off. The body stays in a low grade inflammatory state indefinitely, creating exactly the tissue environment that promotes abnormal cell survival. Curcumin directly inhibits NFKB signaling. It does not vaguely reduce inflammation in an unspecified way. It targets that specific switch and helps turn it off. It also has documented effects on angiogenic signaling and on genes that regulate the cell cycle, meaning it touches all three of our defense systems, though with less potency than the food we will reach at number one. But here's the critical detail that most people miss completely. And missing it means they get essentially none of the benefit. Curcumin is extremely poorly absorbed on its own. When you eat turmeric, your digestive system cannot extract most of what is there. It passes through largely intact. The bioavailability problem is real and significant. The fix is almost embarrassingly simple: black pepper. The active compound in black pepper, called piperine, has been shown in published pharmacokinetic research to increase curcumin absorption by up to 2,000%. Not 20%. Not 200%. 2,000. A study in the journal Planta Medica demonstrated this effect directly, and it has been replicated multiple times since. If you are cooking with turmeric without adding black pepper to the same dish, you are getting a fraction of the possible benefit. Always together, every time, no exceptions. One common mistake beyond the pepper problem: assuming that concentrated supplemental megadoses produce better results than culinary use. The research does not support this. Consistent, moderate daily intake as part of cooking, eggs, soups, curries, rice, anywhere that warm, earthy flavor fits, is what the best outcomes in population data are associated with. Consistency over concentration. Turmeric with black pepper. Daily, that is number three. The gap between number three and number two is significant. Pay attention here. Number two is broccoli sprouts, not broccoli. Sprouts. The distinction is not minor, it is enormous. Here's the misconception. All broccoli family vegetables are essentially equivalent. A serving of broccoli florets and a serving of broccoli sprouts are nutritionally similar. That is completely wrong. And the difference matters more than almost anything else in nutritional science related to cancer defense. Broccoli sprouts contain concentrations of a precursor compound called glucoraphanin that are, depending on the specific study, anywhere from 10 to 100 times higher than what you find in mature broccoli. When you chew broccoli sprouts, an enzyme in the plant called myrosinase activates and converts glucoraphanin into sulforaphane. And sulforaphane is one of the most biologically studied cancer defense compounds in existence. Here is what sulforaphane does that sets it apart from almost everything else on this list. It activates a genetic regulatory pathway called NRF2. NRF2 is sometimes called the master switch for cellular protection, and for good reason. When NRF2 is activated, your cells ramp up production of their own internal protective enzymes that identify and neutralize carcinogens before they reach your DNA, enzymes that support the identification and removal of damaged cells, enzymes that reduce oxidative burden across multiple tissues simultaneously. You are not relying on an external compound to do the protective work. You are activating your cells to produce their own protection from the inside. That is a fundamentally different mechanism, and it is far more robust. A landmark study from Johns Hopkins School of Public Health demonstrated that participants who ate broccoli sprouts daily for 2 weeks showed measurable increases in the activity of multiple protective enzymes in their blood and tissue. The lead researcher, Dr. Paul Talalay, who spent decades studying sulforaphane, described the effect as activating what he called a sustained, long-lasting antioxidant response. Not a short spike, a sustained elevation of your own protective systems. Sulforaphane also has documented effects on epigenetic regulation, specifically the reactivation of tumor suppressor genes that can get silenced over time by a process called DNA methylation. In simple terms, some of the genes whose job is to prevent abnormal cell growth can get switched off as we age. Sulforaphane appears to help switch them back on. Now, the critical practical detail. Preparation destroys everything if you get it wrong. High heat inactivates the myrosinase enzyme. No myrosinase means no sulforaphane conversion. If you cook broccoli sprouts thoroughly, you are eating something that looks like broccoli sprouts but has lost most of its defining biological benefit. Raw is best. A very brief light steam of 1 to 2 minutes is acceptable and may even improve glucoraphanin extraction slightly. Beyond that, you are losing the benefit quickly. Add them raw to salads. Put them on top of sandwiches or bowls after cooking everything else. 2 to 3 tablespoons per day is a meaningful dose. Growing your own takes 5 days from seed to harvest and costs almost nothing. A bag of seeds produces far more sprouts than you can find at most grocery stores for a fraction of the price. One additional technique that many people do not know: if you eat mature cooked broccoli, you can add powdered mustard or prepared mustard to the same dish after cooking. Mustard seed contains its own myrosinase enzyme, and when you combine it with the cooked broccoli, it enables sulforaphane conversion even in the absence of the broccoli's own enzyme. Small details like this are the practical difference between a food that delivers its full benefit and one that delivers a fraction of it. Now we arrive at number one. I need you to stay with me for a moment before I name it, because understanding why it is number one is more important than the name itself. Think about what we have built here. Number six gives you a daily source of anthocyanins that reduce oxidative DNA damage and feed your immune system's first line of operation. Number five adds a mechanism for activating cellular self-cleanup, the apoptotic pathway that removes damaged cells before they multiply. Number four adds specific interference with blood vessel formation, working directly on your angiogenesis defense. Number three turns down the chronic inflammation switch at a genetic signaling level, supporting both immune function and angiogenic regulation. Number two activates your cell's own master protective enzyme system and helps reawaken silenced tumor suppressor genes. Each of these is powerful. Number one is the food that, in the research record, is documented to touch all three defense systems simultaneously with the broadest and most replicated body of evidence of any dietary compound in this list. It does not seem like it should be able to do all of that. When I tell you what it is, your first instinct may be to feel underwhelmed. I am asking you to hold that instinct and actually follow the biology. Number one is green tea. Specifically, a compound inside green tea called EGCG, which stands for epigallocatechin gallate. The name does not matter. What it does is what matters. EGCG is one of the most studied natural biological compounds in modern cancer research, not studied in the casual wellness blog sense, studied in clinical trials, in mechanistic laboratory research, in large population epidemiology. There are currently active clinical trials at major cancer centers investigating EGCG as an adjunct compound in cancer treatment protocols, not as an alternative to medicine, alongside it. Here is what it does to each of our three systems. On immune surveillance, EGCG has been shown to support the activity and proliferation of natural killer cells, the frontline immune cells whose specific job is to identify and eliminate abnormal cells before they can form clusters. A study published in the Journal of Immunology demonstrated enhanced natural killer cell cytotoxicity in participants with elevated green tea intake compared to a control population. The effect is measurable in blood samples. On inflammation regulation, EGCG inhibits multiple inflammatory signaling pathways simultaneously, including NFKB, the same switch curcumin targets, as well as COX2, an enzyme involved in a separate inflammatory cascade. The combination of inhibiting both pathways gives green tea a broader anti inflammatory reach than almost any other single dietary compound. Research published in the journal Cancer Prevention Research found that regular green tea consumers had significantly lower circulating levels of multiple inflammatory markers compared to non consumers across a cohort of over 40,000 participants followed for a decade. On angiogenesis control, this is where green tea's evidence is arguably most striking. Dr. William Lee and colleagues at the Angiogenesis Foundation identified EGCG as one of the most potent natural angiogenesis inhibitors documented in scientific research. Across multiple independent laboratory and clinical studies, EGCG consistently interferes with the signaling molecules that tumors use to stimulate new blood vessel growth. Without those new vessels, even established abnormal cell clusters cannot grow beyond a few millimeters. They starve. Three systems, one compound, measurable effects on all three. Now, here's the population context that reinforces all of this. Studies examining cancer rates in regions of Japan where green tea consumption averages 3 to 5 cups per day and has for generations consistently show significantly lower rates of certain cancers compared to populations with minimal green tea consumption. Stomach, colon, and breast cancer rates in particular have been subjects of active epidemiological research connected to green tea intake. No epidemiological study can prove causation perfectly. There are always confounding variables. But when the population data aligns this consistently with the mechanistic evidence showing exactly how and why EGCG interacts with these specific biological pathways, the convergence is not something to ignore. There is one critical thing to understand about why green tea is different from black tea, even though both come from the same plant. Processing is everything. Black tea leaves undergo oxidation during production. That oxidation converts most of the catechins, including EGCG, into different compounds. Some of those compounds have their own benefits, but the EGCG concentration drops dramatically. Green tea is minimally processed, which is why it retains its catechin profile. If you switch to green tea, expecting the same effect from black tea, you are working with a fundamentally different compound profile. The practical guidance is 2 to 3 cups of properly brewed green tea daily, freshly brewed from whole leaves or high quality bags in hot water. The temperature matters slightly. Water that is too hot, above about 80 degrees Celsius, can degrade some of the catechin content. Steep for 2 to 3 minutes. Hot or cold after brewing is fine. Matcha is a powdered form of green tea where you consume the whole leaf rather than just steeping it. The EGCG content per serving is substantially higher than regular brewed green tea. If you enjoy the flavor, it is an excellent way to increase your intake and get more consistent compound levels. The mistake? Bottled sweetened green tea drinks. Most of these products contain minimal actual green tea and have EGCG levels so low as to be clinically insignificant. They are a flavored sugar beverage wearing health branding. The added sugar actively promotes the chronic inflammation you are trying to reduce. Read labels carefully. If it is sweet, it is not doing what you think it is doing. Green tea, properly brewed, daily. That is number one. Now, I want to pause here because we are about halfway through what I want to share with you today, and I want to check in for a moment. When you think about the information in this video, do you feel more like someone who is taking control of their biology? Or more like someone who is just getting a list of things to add to a grocery order? I ask, because that distinction matters. Leave me a comment with how you are thinking about this. I read them. And the community that is building around this channel is one of the things I am most genuinely proud of. Let us keep going. Now that we have covered all 6 foods, I want to give you the honest context that makes this framework actually useful rather than just informative. First, the gut microbiome layer. The benefits of several foods on this list, particularly pomegranate and broccoli sprouts, are not delivered directly. They are mediated by your gut bacteria. The L-tannins in pomegranate cannot be converted into their most active form without specific bacterial species doing the conversion in your colon. Sulforaphane's downstream effects are also modulated by the microbial environment in your digestive tract. This matters because many people have disrupted gut microbiomes. Antibiotics, chronic stress, diets heavy in processed food, all of these deplete the bacterial communities that activate some of your most powerful dietary compounds. You may be eating the right foods and getting a fraction of the possible benefit because the gut ecosystem needed to process them is not thriving. The good news, the foods on this list are also largely prebiotic. They feed and diversify the bacterial communities that activate their own benefits. Consistent intake improves your capacity to benefit from consistent intake. The effect compounds in the right direction over time. On supplements versus whole foods, I get this question regularly. Supplements can play a supporting role, but the evidence consistently favors whole food sources. A broccoli sprout contains glucoraphanin, myrosinase, fiber, vitamins, and dozens of other compounds that interact with your gut bacteria in ways that a sulforaphane capsule simply cannot replicate. Population studies consistently show stronger outcomes associated with dietary patterns than with isolated supplement use. A high-quality curcumin supplement with piperine is a reasonable choice for someone who cannot cook with turmeric regularly. But it should complement a whole food foundation, not replace it. Third, the question I know a portion of you are carrying. What about people who already have a diagnosis? This is a question that deserves a direct, careful answer. Dietary support during and after cancer treatment is an active and growing area of oncology research. There is meaningful published evidence that anti-inflammatory, polyphenol-rich dietary patterns are associated with better treatment outcomes, improved response to therapy in some cancer types, and reduced recurrence risk following treatment completion. The mechanisms are exactly the ones we have been discussing: supporting immune function during treatment, reducing inflammatory signaling that promotes regrowth, strengthening the biological systems that clear residual abnormal cells after treatment ends. But this must be said clearly. If you are currently in active treatment, you must discuss dietary changes with your oncologist before implementing them. Green tea catechins, specifically EGCG, have documented interactions with certain targeted cancer therapies and can affect drug metabolism in ways that alter treatment efficacy. Curcumin at high supplemental doses can also affect how certain medications are processed by the liver. The general principles of anti-inflammatory dietary support are broadly recommended in integrative oncology. The specific application must be individualized and medically supervised. For people who have completed treatment and are focused on long-term resilience and recurrence prevention, this framework is highly relevant and widely supported by integrative oncology specialists. Now, I want to talk about something I call the cancellation problem because it is genuinely important and rarely addressed in dietary cancer prevention conversations. You can eat every food on this list every single day. You can be consistent and intentional and thoughtful about what you put in your body. And you can still be working against yourself at a biological level if one specific thing is present in your life in excess. That thing is inadequate sleep. A landmark study from the University of California at Berkeley found that a single night of sleep reduced to 4 hours caused a 70% reduction in natural killer cell activity the following day. Not a minor dip, 70%. The immune surveillance system, the one you are supporting with every cup of green tea and every handful of berries, collapsed to less than a third of its functional capacity after one night of poor sleep. That is not a small effect. That is a near total shutdown of one of your three critical defense systems, caused not by food, not by genetics, but by one night of insufficient sleep. Chronic sleep restriction, consistently sleeping less than 7 hours per night, produces sustained elevations in C reactive protein and interleukin 6, the very same inflammatory markers that anti inflammatory dietary patterns are working to reduce. The diet is pushing in one direction, the sleep deprivation is pushing just as hard in the other. Angiogenic signaling is also dysregulated by chronic sleep loss. Growth hormone fluctuations and elevated cortisol associated with poor sleep contribute to a hormonal environment that promotes blood vessel formation in ways that undermine the angiogenesis control you are building through diet. Sleep is not a lifestyle luxury you can trade for productivity. It is a non negotiable biological requirement for the defense systems we have been discussing. 7 to 9 hours per night consistently is not excessive. It is the baseline at which those systems function. Chronic psychological stress operates through a similar mechanism. When the body's stress response is chronically activated, stress hormones suppress immune function, amplify inflammatory signaling, and disrupt the hormonal regulation of cell growth. Dietary polyphenols can partially counteract some of these effects, but they cannot fully compensate for a sustained stress burden that the body was never designed to carry indefinitely. I raise these things not to make this conversation more complicated. I raise them because understanding the complete picture is what allows you to make choices that actually move the needle on your biology. Diet alone, without sleep, and without some management of stress burden, is fighting with one arm. All three together create something qualitatively different. Movement belongs in that same category. Regular, moderate physical activity, around 30 minutes of brisk walking or its equivalent most days, has been shown in multiple studies to increase natural killer cell activity, reduce chronic inflammatory markers, and support the same biological systems as the foods we discussed. There is a specific mechanism worth understanding. Skeletal muscle, when it contracts during exercise, releases compounds called myokines into the bloodstream. These have measurable anti inflammatory effects that compound over time with consistent exercise. Your muscles are not just structural, they are an active part of your immune and inflammatory regulation system. They only release those beneficial compounds when they are used. A daily walk, resistance exercise a few times a week, not for aesthetics. For the myokines, the immune support, and the inflammatory regulation that builds alongside everything else you are doing. Now, let me give you the practical implementation piece because information without application is just entertainment. The principle that works, both from clinical experience and from behavioral research, is addition over subtraction and habit anchoring over willpower. Do not try to change everything at once. You will not sustain it. The research on dietary behavior change is very clear on this point. Start with green tea. It is number one on our list, and it is the most accessible change to implement. If you drink coffee in the morning, keep your coffee. Add 1 cup of properly brewed green tea in the early afternoon as a deliberate ritual. Make it the same time every day. Attach it to something you already do. Within 2 weeks, it is part of your pattern. Once that is established, Add berries to your existing breakfast. On top of yogurt, stirred into oatmeal. As a side, 1 cup. Already bought, already in the refrigerator, no new meal to prepare. Then add turmeric with black pepper to one meal you already cook regularly. Eggs in the morning with a pinch of turmeric and pepper. Soup, rice, stir fry, one dish, one additional ingredient. The inflammatory profile of that meal changes meaningfully. Broccoli sprouts require one small logistical step, keeping them available. Buy a small bag at the start of each week and store it at eye level in your refrigerator. Visual accessibility drives consumption. 2 tablespoons on top of a salad, a sandwich, or a finished bowl. No cooking. No preparation time beyond the opening of a bag. Pomegranate seeds, when they are in season, are one of the great pleasures of eating with this framework. Off season, a small amount of pure, unsweetened pomegranate juice a few times per week is a practical substitute. If you grow them yourself, seeds freeze well. Purple potatoes you rotate in once or twice a week in place of white potatoes. Baked. It requires no additional cooking skill or preparation time. None of this is a diet in the conventional sense. There is no elimination, no restriction, no calorie counting, no phases. It is a series of small additions, each one building on existing habits, that over months creates a genuinely different biological environment inside your body. That last point matters. It takes months, not days, not weeks. The population studies that show the clearest benefits are measuring people who have maintained these dietary patterns for years. The biology does not transform overnight, but consistent input produces measurable change at every milestone. And the earlier you begin, the longer the compounding works in your favor. I want to close with something I think is important beyond the biology. There is a dominant narrative in health media that frames your relationship with your body primarily as a defensive one. You are protecting yourself from threats. You are trying not to get sick. You are avoiding, restricting, monitoring, waiting for something to go wrong. That framework is exhausting. And from what I have seen clinically, it does not actually produce the most resilient people. It produces anxious ones. What I want you to take away from this video is a different orientation entirely. Your body is not waiting to fail. It is not a passive system hoping you make the right choices. It is an extraordinarily sophisticated defense operation that is actively working on your behalf every single second, with or without your conscious participation. What you are doing by implementing these dietary changes is not protecting a fragile system. You are strengthening a powerful one. Every cup of green tea is reinforcing your immune surveillance network. Every handful of broccoli sprouts is activating your cell's own protective enzyme machinery. Every meal made with turmeric and black pepper is helping turn down the chronic inflammation that slowly shifts your biological terrain in the wrong direction. That is not fear. That is investment. That is a relationship with your own biology built on understanding and on respect for what your body is already doing. And it begins with the very next thing you eat. If this video gave you something genuinely useful, I hope you will subscribe. Not because I am trying to build an audience for its own sake, but because what I am working on here is a sustained, science-grounded conversation about how to build biological resilience across every year of your life. That is a conversation worth continuing. There is one specific daily habit that quietly cancels out many of the protective effects we discussed today. It is present in the lives of most people watching this right now, and almost none of them know it is working against them. That is the subject of the next video. It will be worth your time. If you want the full implementation framework, including a week-by-week approach to building these habits and the science behind the specific compound interactions we did not have time to cover here, the longevity guide linked in the description goes into significantly more depth. Take care of yourself. I will see you in the next one.